like I do, but I do, Lord, I do.
Hello, everyone. Please, if you can come back in, and those in the room, if you could please take your seat back. And we're just about to start in 23 seconds.
23 seconds. Is that what we said? All right. All right, we're going to get going. And I'm going to speak very loud that we're going to get going so that people out there can hear us that we're going to get going. So welcome to everyone who came to this session, and welcome back from the break, as well as those on Facebook Live. And we will be doing a small opening to this session in a moment. But first, I'd like to welcome Jean to speak to a little bit of an intro on her own.
Thank you so much to the Alliance for scheduling this talk, and for the wonderful panelists for being here and taking their time to talk about this very interesting and important topic. Before we start really getting into it, I will just share a little bit about the impact of genetic ALS in one family in one generation. This is my mother's generation. They witnessed their grandfather die of ALS in their youth in 1970, and their mother die of ALS in their young adulthood in 1990. Then there was quiet for 27 years, aside from anxiety and fear. So right here, this is my mother, Kathleen. She was diagnosed in 2017 at age 68. She was always open about her fear of developing ALS with her family, with strangers, and of course with her doctors. It still took a year to receive an ALS diagnosis after progressive paralysis started. She pounded the table in the neurologist's office, insisting that she had ALS in January 2017, and the neurologist dismissed her family history, called her 2 drop feet, peripheral neuropathy unexplained, and she would not be diagnosed until she also lost use of her hand and started slurring her words. After my mother's death, my Aunt Joanne was diagnosed at age 58. She had been symptomatic for at least a year before she allowed herself to be seen at the ALS Families Project at Columbia under the auspices of being an at-risk person, but of course, she walked out of that appointment with an ALS diagnosis. She died in 2023. She was the youngest of the generation to make it to adulthood. They had a sister who died tragically in childhood, and my mother was the oldest, and my aunt was the youngest, and there were 5 in between waiting to see who was next. And that was this year. My Uncle William was diagnosed at 74. He had been progressively frail for many months before his diagnosis and was unable to do his post-retirement job as a dog walker. He is still battling ALS right now. My brother just brought to him the ramps my grandfather procured for my grandmother, which my mom kept a hold of for her own eventual use, and now he's getting to use them. So in this one family in one generation, not a year has gone by without a member suffering from ALS for the last 7 years since 2017, though due to the rapidity of the disease progression and the delayed diagnosis, despite the known risk, none of the 3 had an ALS diagnosis at the same time. And this is repeated across many families across the world. Thank you.
Thank you, Jean. So I want to say thank you to the Alliance for this panel and for having the good fortune of being the moderator of this panel. My name is David Taylor. I'm the VP of Research and Strategic Partnerships at ALS Canada, and this panel is about the path to pre-symptomatic ALS MND trials. There are a lot of researchers who believe that understanding the biological disease of ALS upstream of symptom onset is going to be our best opportunity and chance to be able to make a headway towards substantial treatments for the disease and maybe someday a cure for the disease. But it is really complex. And so when we think about how that's going to happen and how do we coordinate that around the world, we should first take a step back and take that first step towards explaining it to an audience at the Alliance about what some of those challenges and hurdles are. And when you think about what does that mean, the biological disease upstream of symptom onset, many people think of the disease as starting at symptom onset. Well, it was actually visionary science and work done by one of our panel members more than 15 years ago to start working with individuals who are at high genetic risk to understand the disease that is happening upstream of that symptom onset that allowed us to have a panel like this today in 2024. And it is the need to be able to step into that today that will eventually allow us to understand that disease upstream of symptom onset and to be able to get to those treatments faster in the future. And so there is no better panel that you could find in the world for this, representing academia, the ALS community, and industry to be able to speak to this important topic. And it's something that I suspect will be coming up at Alliance meetings year after year, so we can look at this as some of the first steps into that. We're going to hear probably some of the challenges that are with exploring asymptomatic, pre-symptomatic clinical trials for ALS MND, and we're going to hear some of the boundaries that should be pushed, and I think that's what makes for such a great panel. So to introduce our panel on the academic side, first we have Professor of Neurology and of Public Health Sciences at University of Miami Miller School of Medicine, Dr. Michael Benatar. Dr. Benatar is that individual I talked about who had that visionary idea more than 15 years ago that has really paved the way for these conversations we're having today. Next to Dr. Benatar, needs no introduction, Gene Swidler is the Executive Director of Genetic ALS and FTD and the Legacy. Only in the past few years has become an absolute tour de force when it comes to advocating for people at high genetic risk. And her acumen at being able to understand the literature, to meet people where they're at, to show an incredible ability to integrate what she wants to do to push those boundaries, but at the right way, that has put her at this table today, and we're so grateful that she's here. And next to Jean is Stephanie Fredette. Stephanie is the Vice President, Head of Neuromuscular Clinical Development at Biogen. And she'll hate that I'm going to say if you all are very aware of tofersen Qalsody being the real universally accepted way that we can now understand that ALS is a treatable disease, there is likely no one in this world who has put in more effort to get that to people than Stephanie Fredette, and we are very grateful, and she's probably one of the most respected people in industry in ALS as well. So this is a really awesome panel. So we're going to start, and we're going to give each of the individuals, the panelists, a couple of minutes, probably about 5 minutes each, to sort of talk about what would be that first bit of conversation you want to talk about. What are the challenges and hurdles that we face in terms of approaching asymptomatic, pre-symptomatic clinical trials, some of the things that are already going on we'll hear about, and we'll go from there. And then please do think about questions, because we're going to open Q&A for this discussion after we have these opening statements. So please, passing it to you, Dr. Benatar.
Great, thank you, David. Is the audio okay? Great. So, I think this panel is entitled The Path to Pre-Symptomatic ALS MND Trials. And in reflecting on that, I think it's helpful to look forward, but it's also helpful to look past. And maybe to underscore that for unaffected carriers of SOD1 mutations, that future is now. That trial is now ongoing. But it's helpful to understand how it is that we got there. This trial, I think, has been possible because of some of the foundational work that we had done through the pre-FAL study, trying to understand the pre-symptomatic course of disease, identify biomarkers that might tell us when disease is coming, and then through a fantastic partnership with Biogen, as tofersen emerged over the horizon as a safe therapy, as an effective therapy for people with manifest SOD1 disease, we were ready to say, let's put these knowledge pieces together and design a pre-symptomatic trial that might aim to prevent disease. And the key thing is that when we started pre-FALs back in 2007, we had this idea of prevention in mind, but we knew that there was much more that we didn't know than we did know that would be essential for us to know if we were ever going to do this sort of study. And the key was to say, let's not wait until we have a potential therapeutic that's worthy to study in mind before we lay that foundation. And so laying that foundation meant that when the drug candidate emerged, we were ready to go. And as ready as I think we were for this subset of the SOD1 population, I don't think we're quite ready for other genetic forms of disease, and certainly not for non-genetic forms of disease. But we need to be learning from that experience of laying that foundation now so that when those therapeutic candidates emerge, we're ready to go. So we can come back and talk more about that, but maybe just to say for the most common genetic cause of ALS, the C9orf72 repeat expansion, as this group well know, people are at risk not just for ALS, but also for frontotemporal dementia, and that's true of other genetic causes of disease. So when we're thinking about pre-symptomatic trials, we have to think about what are we preventing, and it's not just ALS. We're also trying to prevent FTD, and really any phenotypic manifestation of what that genetic risk in this case, or risk, might put somebody at risk for. And to your point, David, that's why we have to understand the biology and develop treatments that target that biology. And to the extent that we do that, we'll be working towards preventing not a single phenotype, but any phenotype associated with that underlying cause. But I do want to underscore as well, so I realize I was supposed to ask you to put up a slide. I forgot. So there's a slide if anybody wants to follow along, so we'll bring that up. But I do want to say that I think, and I'm hoping this panel will go beyond genetics as well. I want us to open up our minds to the possibility that we should be thinking towards the future of pre-symptomatic studies and prevention trials in a non-genetic population or a non-genetically at-risk population. And the way to do that is that we have to identify the risk factors for the non-genetic forms of disease. The reason we started with the genetics is because that was the tractable place where we could start, where we could get a handle on studying people at risk for disease. And what we surmised, and I think we now know, is that lessons that we learn will have broader relevance. It won't be the same solutions, the same tools for every case, so for every genetic form or non-genetic form, but the principles will apply, and they're things that we can learn. And maybe the last thing to say is that success now and success going forward, I think, is critically dependent on partnerships. It requires partnerships between the ALS community, the FTD community, and other disease communities. It requires partnerships between academia and industry. It requires partnerships that engage stakeholders across the spectrum, the carrier community, the regulatory agencies, academia, industry, all of those. And so I think as we're looking towards that future, I just want to underscore the critical importance of those partnerships. And I think we'll come back and talk about many of these things, but I'll stop there.
And Jean?
Thank you so much, Dr. Benatar, for laying all that out. I'm so thrilled that there's brilliant, compassionate, dedicated folks like yourself and Dr. Fredette working on this, so thank you again. I think my brief comments here are going to definitely be more of a general position on these things, so forgive me. So in my opening, I shared the nature of the existential risk in my family, and that exists in so many other families, and that's an important thing that is not great. But I will stress the need that not just for our families, but for the entire ALS community, testing drugs earlier in the disease process is important for us to try to make progress in this terrible disease. Because we have had failure after failure after failure after supposed success, which was really a failure in drug development for people impacted by TDP-43 ALS, which is 97% of ALS, which is all of sporadic and most of genetic, including C9. And we really need to cure this disease and make huge impacts on disease progression along the way. Testing drugs earlier in the disease process, which is currently only practical in those with a high genetic risk, would allow for more definitive answers if any possible therapeutic approach works better or only earlier. And while we may be tempted to think only of possible pre-symptomatic trialing of drugs as standalone trials like the ATLAS trial for SOD1 that's happening right now, The most practical path forward for beginning to learn about the impact of possible approaches earlier is using smaller cohorts of genetic carriers with some confirmation of biological disease in otherwise larger studies of drugs in symptomatic ALS patients. This is a tried and true approach in rare drug development, and cohorts and trials that do not contribute to the pivotal clinical efficacy question can help with questions of accelerated approval or guide follow-up trials. We only need to look at the VALOR study for this example, where the pivotal question was the impact of the drug on a fast-progressing subset of the full trial cohort. While that pivotal clinical efficacy question did not meet its threshold, the trial received accelerated approval based on the NFL response in the full cohort. So the patients in the study who were not part of the main efficacy question contributed to the accelerated approval. So let's follow that example in all late-stage ALS trials. And then a few loose notes. One is about the FDA. The FDA has made clear that they support experimenting in earlier cohorts. They've made lots of statements and many meetings about this. And then also, they have approved trials with pre-symptomatic cohorts with biological disease activity in progranulin FTD and in a small phase 2 with C9orf72 carriers for baricitinib at MGH. But I realize the willingness of the regulator is not the only issue here. And secondly, the group that we are considering for this are not healthy people. They might be fully functional people, but we should only be considering trials in people with signs of biological disease as an inclusion criteria for it. It has been established for many decades that motor neurons are extensively damaged before any functional impairment is reached, and this may be the reason drug development has been so underwhelming in ALS over the years. Just briefly to speak to safety, certainly for novel medications with uncertain side effects, establishing some initial safety in symptomatic patients seems reasonable. So really just looking at late stage larger trials with smaller genetic cohorts would be what might make sense. And finally, for biological signs of disease, it may be true that much more is needed to have full confidence in what the biological trajectories mean. However, if uniform biological response is seen at the levels observed in VALOR in a biologically active pre-symptomatic cohort, that would be compelling data that could be created to support a full package for the FDA combined with positive outcomes in a fully symptomatic cohort, or clear need for a pre-symptomatic confirmatory trial if there is a separation in the response between those earlier and later in the disease. Thank you so much.
Thank you, Jean. I'm going to break the microphone before I start. I asked Dave if I could go last because I see the world very similarly to Dr. Benatar and Jean, so I didn't want to I won't be redundant, but just a couple of thoughts. I think, the Atlas study, aside from my children, is probably the thing that keeps me up the most nights. It's a tremendous honor to be part of running this study, but it's also a tremendous responsibility. And the learnings that we are accumulating in real time are something that we're continuously trying to share with the world, because while this study, and we've alluded to this, but is in a specific subset of the SOD1 population, faster progressing variant carriers, of SOD1 variants. And while it's specific to that population, the learnings from the study, of course, are sort of paving the way for how to do this going forward. But as Dr. Benatar alluded to, we can only do that if we understand what are those factors that are moving earlier in the disease process in other subsets of the population. And we were only, as both Dave and Dr. Benatar said, we were only able to run the Atlas study because of our understanding of what was occurring in these at-risk, quickly progressing SOD1 variant carriers in advance. It's the only reason we could get buy-in from regulators to run the Atlas study. And so we have a lot more work to do to understand these factors and like I said, what is moving in advance of clinically manifest disease in the broader ALS population. So everything we can do to generate and build that understanding. I know there's a lot of ongoing work across the globe to characterize the natural history in these at-risk carriers, and that work is absolutely imperative. I think the Atlas study will teach us a lot about tofersen, but what excites me most is what it will teach us more broadly about the disease. We talk about sort of the fact that we will have CSF samples from these at-risk carriers prior to elevations in neurofilament. So when they're disease process has not yet started. What will we learn there? What will we learn on comprehensive EMGs in those individuals? How will that differ at the time that neurofilament levels have gone up? And so some of these factors, if we're looking at levels of SOD1 protein and things of that nature, may be specific to the subset of the population that we're evaluating in ATLAS, but many of these factors will not be. One of the perhaps most important things that we're experiencing and learning and need to do more of is, there's a recent publication that both Jean and Dr. Benatar were on about monitoring of at-risk carriers in the real-world setting. Because one of those things that keeps me up at night is as we have individuals that are not eligible for ATLAS or that are part of ATLAS but don't ultimately make their way into the interventional study period, then what for those individuals? So the more that we can have global partnerships to ensure that those individuals are getting the monitoring that they want and need, the better. And if we can learn from that experience, then I think we all win. I think that one other thing that I'll mention is, and this has been alluded to by all of the panelists, we run the risk that we are intervening too late. We recently had an Ataxin-2 readout, and there are a number of different reasons why that ASO may have failed. One of the hypotheses is that we intervened too late. The preclinical data that we have on Ataxin-2 are primarily in the pre-symptomatic setting. And so if we could intervene earlier with some of these targets, maybe not pre-symptomatically, but even at the time of early symptom onset, are we going to be able to more robustly test these scientific hypotheses and remove one of the most important confounders, because we know time matters in these individuals. So while we're focused on pre-symptomatic intervention, we're also needing to do more to understand those early clinical signs and symptoms, and this has been a focus of Dr. Benatar as well. So I think the more we can define that early symptomatic period, the more we can enrich clinical trials, particularly if we have, as Jean alluded to, objective biomarkers that aren't making us dependent on progression clinically. We can run different clinical trials and be more inclusive in those clinical trials for the broader population as well. So just a couple of thoughts to touch on.
Please, if you'd like to, before we get into any questions, sure.
Yeah, I just wanted to add a comment and a thought. I think, when we're trying to identify an effective therapeutic, we have to have a signal that can tell us that that therapeutic is meaningfully effective. And the catch-22 that we're in is that we can measure that therapeutic signal more readily in the affected population, but it's also the population in which the disease may be too advanced for the therapeutic to work. And when we're in the pre-symptomatic population, until we have those good biomarkers that are something we can measure biologically, we don't have anything that we can measure, and so we can't judge and evaluate whether a therapeutic is having an effect. And so maybe one of the most essential keys to get us to a future of more pre-symptomatic trials is development of quantifiable biomarkers in the pre-symptomatic state that we can use to tell us, is the drug having an effect? And whether that's, for example, including carriers, unaffected carriers in a clinical trial primarily focused on the affected population, we still need to be able to measure that effect, and the biomarker will be key.
And so I just wanted to also level set for everyone in the audience. When we think about the trial that's been talked about here, the landmark ongoing ATLAS trial that's happening, at a very basic level, when we're thinking about this, from the work that Dr. Benatar has done, it has shown that people with a specific subset of variants with SOD1 variants will typically convert to having symptoms in approximately 1 year. And I know that's a very crude overlay of saying it, but the idea here is that if they are able to be treated with a therapy that we know is effective that far in advance, when there's a signal of a spike in neurofilament, which is representative of neuronal damage, there's the opportunity or the desire that this may actually have people going well past the time when they typically would have got the disease. And of course, we all hope that this may show that people who have a history of fast progressing disease may not get the disease. And that is what we're talking about here, which is incredible. And something 5 years ago, just you would have conceived of it but a lot of us would never have done that. So I think that for an inspirational piece, that's really why we're up here, is to think about how we can do that for everybody. And as you said, even the non-genetic, or at least non-obvious genetic individuals with the disease. Before we get into the questions for the audience, and please do think about your questions, I just have one additional question here for the panel, and that is, okay, so we know that for other individuals at high genetic risk, or even those who are more complex with terms or with regard to how their disease starts, what would you say the research community, given that there's a lot of organizations supporting research out here, should be doing globally, collaboratively, to try to support doing this as quickly as possible and understanding the complexities of the other forms that we can try to approach in the same way? And then, of course, I would also like to add to that, if Jean could talk about, as we get to those things that we should be doing, what should we be mindful of for the community who we might be having volunteer for a lot of those studies to effectively do it in the best way? So perhaps, Michael, you had said it's complicated. Perhaps you could help us understand what we could be doing.
Yeah. Maybe I can sort of use an example to try to illustrate a broader point. I think we need, if we're going to understand, if we're going to have a future of pre-symptomatic trials, let's at least say for now in other genetic populations, we need to study the carrier community at scale. But it's not just at scale, it's also with the relevant depth and care of clinical phenotyping and biological phenotyping is the wrong word, but biological understanding. We want speed and we want scale, but not at the cost of quality. Because I worry that if we run too quickly, we run the risk of investing an enormous amount of time, energy, resources, and then we turn around and we say we don't have the answers that we need. So for example, we've been able to identify prodromal stage of ALS that we've called mild motor impairment, which to this community will come as no surprise that developing overt ALS is not like flicking a light switch. It's not like it's not there and then it's there. Right? It's more like a dimmer switch is the analogy I give people. Biology is a continuum. The disease is gradually emerging, and then it sort of appears above the clinical horizon and becomes clinically apparent. We didn't know that when we started, and for years I was scratching my head saying, I no longer think I know what ALS is, because I'm seeing all of these, in scare quotes, abnormalities, but I don't know what they are. This is not ALS. And it was only through the careful approach to phenotyping that we could identify a prodromal state. Now, to the extent that we're right about that, maybe that then becomes studyable in other genetic populations and even non-genetic populations. I'm thinking here by analogy to mild cognitive impairment in the Alzheimer's world, and our clinical trials in the Alzheimer's world have moved into that earlier prodromal stage of disease. And so we didn't set about to discover a prodromal stage, but we set ourselves up to be able to identify early markers through a careful approach to phenotyping, and knowing that disease could emerge in an arm, in a leg, the right, the left, the bulbar, the respiratory apparatus. And so we knew we had to broadly phenotype to understand that. The same is true if we're looking biologically. We don't know where that signal's going to come. Is it going to be blood, plasma, serum, RNA, CSF, DNA, urine? So we have to think broadly in terms of setting ourselves up for that success. And so I guess what I'm trying to say is we want to do this at scale and we want to do it quickly, but I want to make sure that we collectively think about what we need to set ourselves up for success.
And I'll just double click on 2 points that Dr. Benatar mentioned. One is sort of the scale bit, which I think what you have done at the University of Miami is unbelievable. The ability of other individual institutions to do the same thing is inevitably going to be more limited for the broader population. So the more that groups are coming together and joining data sets and testing samples with a single assay versus everyone having their own assays that they're running where we can't actually integrate data across these different studies. So the work that Precision and AMP-ALS are doing and many others like it, I think is imperative to get to scale. And then to Dr. Benatar's point about quality is also frequency. And that's something that we're learning in the context of the Atlas study, is the importance, although it can be burdensome, of having frequent data collection points to really understand and characterize this early part of the disease process. And so we have to figure out what that ideal interval is, but it is, it is something that we have to consider more broadly.
So just to jump back in, thank you guys so much for your well-reasoned and argued thoughts. I just wanted to stress that right now there's drugs being trialed in ALS where the outcome in the fully symptomatic cohorts are leading the field to exclude hypotheses of what the disease is, and without testing earlier, being as a hedge to show that that's not what the issue was. And there are, something like a few thousand C9 carriers developing ALS or FTD every year in the US, more globally. And there is published evidence from the FTD cohorts that there's a 3-year a significant separation from controls for C9 carriers who are going to develop disease 3 years previously. And I realize that's not really ready for prime time for like a full accelerated approval, most likely for C9. But if there's people in that position with an elevated NFL that is significantly different than controls and age-matched controls and all of this, and there's drugs that need to be trialed, and we can find 20 or 30 or 50 of them with proper disclosure of their NFL results, just to see if there's some huge difference in the biological response in the symptomatic versus the earlier people, that would tell us something. If there's 30 people with a uniform response, that might not be statistically significant to say that it definitely works, but it would be a clear signal that there's something there that needs to be pursued, whereas right now, none of that's happening. So that's kind of where I would just frame it.
Yeah, perhaps I can just say, I partially agree with you. The devil is in the details. So the all FTD data is very interesting. But what they show is that at a group level, on average, neurofilament levels are higher in the carrier population than a non-carrier population. That's quite different from being able to say, this individual, their neurofilament is, quote unquote, elevated. So showing a group difference is not the same as showing that an individual is elevated. And so if one wants to use that as a response marker to know whether you've impacted that, you have to know that the people you're enrolling, that they individually are at risk of whatever that outcome is, or that there's a measurable substrate to understand. So I agree with you, but I also worry that we might run before we walk with some of those data because of the group versus the individual level interpretation. And I think that also depends on how we then use that information. If it's to sort of inform a broad understanding of biology, or if it's to inform selection of individuals to be enrolled in a trial, those are sort of different utilizations of that information. And I think it can lead us in different ways, in different directions.
Thank you so much. I just want to affirm I completely rely on you and your colleagues to put the fine point on the details here, but there's just a lot coming out about imaging markers or connectivity markers and blood markers and stuff. There's things that could be looked at, and if there is no response on those markers, I think that's probably a sign that it's not a home run. That's not to say that it wouldn't be something at the level of a riluzole or something, but for a huge effect, we would expect to see a very clear and dramatic biological effect. So just some thoughts.
Yeah, and I think, sorry, Dave,
No,
I know you want to move on. I agree with both of you. I think that the bar for incorporating a biomarker or imaging marker for determination of eligibility is much higher than I realized when we were designing the Atlas study. But I think that's why understanding sort of the natural history is so important. And I don't think we're that far away from trying to enrich a cohort. To state the obvious, we need a drug that then impacts that marker, which is the other part of the equation that I think we're all working towards, is having a drug that can meaningfully affect objective markers of the disease process such that we can make an informed decision. But it's something that we think about all the time. And to be completely frank and transparent, it's something that we contemplated for, had we moved forward on the Ataxin-2 program, for example, is there a way of looking at a C9 cohort earlier in the disease process in more of an exploratory manner as part of that pivotal study? So it is something that we're actively contemplating, and I think we have to zero in on how to meet that threshold that inevitably regulators will require for identifying the population.
Just point of order, there was, this is me merely as an observer, there was this baricitinib study last year at MGH where they had an inclusion criteria of some inflammatory markers for 5 pre-symptomatic C9 carriers. I don't understand. I have never seen anything clearly published about inflammatory markers for pre-symptomatic conversion, but the FDA let them do it. So just raising that up.
So I want to, if anyone has a question, please raise their hand. But I want to also, what you're seeing here is incredible dialogue over a very complex situation. And I think that's what's amazing here. And it might be unlike other situations where Michael and Steph and Jean talk about this a lot with each other. And what you're seeing is a snapshot of the conversations that they're constantly having between how do we do this appropriately? How do we meet the urgency that we need to be able to push the boundaries like Jean's talking about, and how do we integrate with industry to be able to make this actionable? So, I mean, I find it fascinating. We couldn't even get to half of the questions that I had written down, but that shows you why what I was saying is we're just in the very, very opening steps of starting to have these conversations, and having them ongoing is incredibly exciting and hopeful, but there's a lot to be done to figure out what that happy medium is between doing it right, but also the urgency that is needed and the care that's needed in doing it. Because Jean, I know you and I had conversations in the past where you explained to me, and that these studies also need to have give back for the communities who are going to be participating in them, and that comes back to your regular monitoring and pushing boundaries and thinking outside the box and all of that. And I wanted to make sure that that gets highlighted too, because it's absolutely critical that we're not talking about people at high genetic risk as just responsible for us to figure out the rest of the disease. And you've definitely learned me much on that over the years, and I appreciate it very much. Does anyone have any questions in the audience? Or we'll just all keep talking up here for...
We really like each other, by the way, just in case that wasn't obvious. We're learning from each other as we're talking.
This is healthy academic dialogue, which is incredible.
Yeah,
there any
I
questions?
have a question here.
Yes.
Yeah, my name is Debbie Lauer, and Dr. Benatar, I first got to listen to you in 2013 at ALSA when you did a familial ALS presentation. And I am so excited to see the progress over these last 11 years. And when I see, the idea of doing a trial for C9, that's what my family has. And I think about what Allison just talked about, that you can go out to rural, in my case, America, and my family, it puts their head in the sand when it comes to, trials and studies and that sort of thing. I have over 120 living descendants from my grandparents. So it took 5 of 8 in my mother's generation. We've already lost 2 in my generation. Now number 3 was just diagnosed, and number 4 will be later this month. So do you have any advice for someone like me who's tried desperately to get my family engaged? I've met with them 4 times over the last decade to teach them what to look for and all about studies and trials, and I'm not getting through. So I would love to know if you have any advice for how to break down that barrier that Allison talked to us about. Thank you.
So thanks for the question. I'm happy to start, but my perspective is only a very small part of this. So I guess my perspective from PREFALS is that when we started, we wanted to be as broadly inclusive as we could and enable people to engage in the way that they were comfortable. So what that meant is we offered people could join the study if they had undergone testing and knew they carried a gene. We offered people who wanted to participate and learn if they go through pre-symptomatic testing and learn that. We also said, if you're just from a family and you want to participate, but you don't want to go through genetic testing, That's okay. We'll do that on the side, and only our stats people will know about that. So we want it to be broadly inclusive. So I guess that's the one message, is as we're designing studies, we should try to embrace as broad a segment of the community as possible. What we've also seen when we transitioned from not only running pre-phals, but then to enrolling for Atlas, is now the motivation changed for many. And so now people said it's not just an observational study, it's also an interventional study, and that brought in an additional segment of that carrier community. And it took some of the people who had decided they wanted what we call non-disclosure to participate without learning genetic results to say, now's the time for me to learn. And so I don't know that I have a clear answer or one size fits all, but maybe to the extent that it's true, and it should be, to say that the research community is looking to engage you on the terms that you want to be engaged on, and it's our responsibility as a research community to rise up to meet that challenge and to find ways for people who want to participate at whatever level, whether it's a remote visit once a year, whether it's an in-person visit every 3 months, whatever it is, we can tailor that to enable as broad a segment to participate. But I'm sure others will have other perspectives.
Well, Debbie's a friend, so we talk all the time, so I'll let Steph go.
I think your perspective is spot on. I mean, I don't have anything much to add. I think that different motivations will be relevant for different people. I think if there's anything that we can do to help educate and make people aware of the fact that we would not be running this Atlas trial without the contributions of the pre-phals participants. It would not be happening. Whatever we can do to shout from the rooftops to help, others understand that, that reliance, I think we'd be very happy to do that, but it goes without saying that we wouldn't be where we are. We wouldn't be having this conversation or running that trial without those individuals and their contributions. So.
And maybe just to add one other thought is to encourage your family members and that broader community to reach out to researchers and learn about whether there's an opportunity to engage on their terms. Not to prejudge it and say that it's going to require X or Y or Z. And certainly if they reach out to us, if we don't have an opportunity for them to engage, we will try to help connect them with other groups where there might be opportunities. So I think to the extent that you can help get them to over that initial hump and just some contact and discussion, then we can try to meet them where they want to be.
And more for the audience, Debbie's heard this, but thank you, Dr. Benatar, for leading the project with Dr. Hyman Patterson about the guidance that just got released for the very first time, which tells people that there's some little things that they should consider doing for their own personal health while they're at risk, which never existed before. So that's new information that we have to make sure that any family is aware is out there if they want to learn about it.
And maybe to say that's another opportunity. If the carrier community starts coming to have their care needs met, maybe that creates an opportunity to learn about research opportunities. We see this in the clinically affected population. They come to multidisciplinary clinics and they learn about ongoing trials and research opportunities. To the extent that we start to build a network of clinics and clinical service for the carrier community, maybe that creates another opening or opportunity.
Great. We have time for one last question.
Okay, I'm going to rock your world for a second. And I'm friends with Debbie too, so this has nothing to do with it. But this is exactly the bias that we uncovered on our trip, is we assumed everybody wanted to know that information. And from an ethical perspective, once you take away their piece of not knowing it, they can never get that back. And so where do you cross that line to say, these people don't want to live in fear and they don't want to know, and that's going to be okay?
Allison, just to be clear, if they are putting their fingers in their ears, they're living in fear if they're telling you that. And we're not talking about Dr. Benatar or Steph, Dr. Fredette going out to these people and telling them that there's new guidance. We're talking about families educating families that there's new information. And people in their own families can judge what is best appropriate to share with their family members.
Yeah.
It was an exact family where the kids did not want to know, and they'd lost their mom, their uncle. They knew that there was a genetic component. They didn't want to know about genetics.
But Allison, that was their family's business. I understand that
Yes, that's what I'm saying too, is that we need to ethically let people live in peace if they value that too.
Yes, Alison, one thought. One of the things we learned along the way when we first started PreFals, and when we were doing this back in 2007, the idea of doing telephone genetic counseling was almost unheard of. I had to strong-arm my genetic counselor into doing this with me. But one of the things, we started from the Huntington's model, where we said we have to give people pre-test counseling and post-test counseling. Pre-test deciding whether you're ready to receive genetic results, then you have a test and you come back and you get post-test. And one of the things we learned is that we needed to introduce a third counseling session, what we call pre-decision counseling. So somebody is just beginning to toy with the idea. They don't know whether they want to engage in this. Let's have a discussion about what it means, what that information would mean to you, to your future. And then if they then ponder that and say, I would now like to go forward, we would then still engage in a traditional pre-test counseling. And so I think that was one of the things we heard and we learned, is that we needed to give people sort of an extra step along that journey and an opportunity to change their minds at any point, and to underscore that you can change your mind at any point until you actually get the result. And once you have the result, there's no going back, right? But you can be sitting down for your post-test counseling and say, I don't want to know, and that's okay.
And we're going to have to cut it there, but thank you very much. As you can see, there's so much to talk about, and I hope the Alliance continues to make space for this very, very important topic. And thank you so much to the panel. Please join me in saying thank you to them. And thank you to everyone on Facebook Live. We'll be cutting off now, and I'll pass it back to Richard.
Whew.
Okay, so, it's hard to follow. This was pretty interesting discussion, an important discussion. So we move to the next panel. I think the pause is good enough. So we need to pause a little we're good? Okay, so to get the streaming off. All right, so we're good to go. I guess our last panel discussion for today is leading us to the Scientific Advisory Council update. So there is a very hard core of the scientific part. And so we have a sort of fantastic panel for that today, and I will invite them on the stage. And I believe Dr. Taylor will also lead the discussion. So Dr. Nicholas Cole, Dr. Kuldip Devi, Dr. Martina De Mayo, again, so you're busy today, Dr. Janine Heckman, Dr. Beck Sheehan, and Dave again. And Dave, please take the lead. Okay, all right, that's good. Excellent, that's better.
Should go before. Okay. Musical chair. Is there enough?
Okay, thank you everybody for staying with us. So my name's Dr. Nick Cole from the MND Association in the UK, and I'm the chair of the Scientific Advisory Council. So the Scientific Advisory Council is a centralized resource, and really our role is to review and provide a global perspective on scientific and biomedical announcements, information and opportunities relating to ALS, MND. So what that really means is we kind of digest the information that's out there and try to put it in a form that's understandable to advise the community. So we're here for you. If you have any questions or you need to understand anything in more detail, you can always get in touch with us. And what we're going to do today is have an introduction to members of the panel, and then we'll open the floor to any questions you might want to ask us going forward. So the Scientific Advisory Council consists of 13 members. Obviously, not everybody's here at the moment, and we have representatives from all over the world, including Colombia, China, Sweden, and Italy, and the USA and South Africa, many places as well. And so we'll just introduce the panel members. So if you could, we'll go down the line, introduce yourselves, and just give us a brief bio of who you are. Over to you, Beck.
So I'm Beck Sheehan. I am the Research Director at Fight MND in Australia, so down in Melbourne. A bit about me, I guess my background is in preclinical MND research, so I was in an MND research lab working on drug treatments in cell and animal models of MND. And then about 8 years ago, I met one of the founders at Fight MND and was really inspired to hang up my lab coat and join the organisation. So I've been with Fight for 8 years. I look after oversee all of our investments in research and care. And I'm really passionate about including the lived experience voice in research. So I've been really grateful through my role to be able to be involved in a number of great initiatives that are bringing researchers and people with lived experience together.
Thank you. Janine.
I'm Janine Heckman. I'm a neurologist from Cape Town, South Africa, and I've already introduced myself as the part of the ALS Africa Research Network. And yes, I'm very happy to represent Africa on the SAC.
Thank you. Martina.
You must be tired of hearing from me. So I'm Martina De Mayo. I am the Scientific Director for the International Alliance. My background, I'm a neuroscientist and researcher by training, worked in the ALS and MND space for almost 15 years now with my PhD and postdoc, and then with some industry experience, and I'm very excited to be here.
And I'm David Taylor. I'm the Vice President of Research and Strategic Partnerships at ALS Canada for the past 12 plus years. And I'd just like to say I've been in ALS for about 24 years, and it is my everyday passion. And I'm just so grateful to be a part of this community. And I say thank you for that opportunity.
Hi, everyone. Kaldab Dave, Senior Vice President of Research at the ALS Association. I'm a pharmacologist by training. Been at the association for over 5 years now, and happy to be on the panel.
Thank you. So I thought we could just start with a few minutes on things that we think are exciting and quite challenging in our own areas at the moment, and we'll just move down the line before we take questions from the floor. So something that I'm particularly excited about in the UK is the start of this pre-trial platform called Experts ALS, which is a way of really taking a small number of people with motor neuron disease and to take drugs. We have lots of potential therapies and compounds, and we need to prioritize which ones could move forward into larger clinical trials. So the principle behind Experts ALS is to measure the neurofilament levels in a small number of people before and after giving an indication to see whether or not the drugs might be working before they move them into a larger clinical trial. So that's something that's just starting off in the UK at the moment, which is quite a novel and new approach, which we're very interested to see how that turns out. Beck?
Great. So some of the things that I think are really exciting in Australia at the moment, we have in Melbourne an MND drug screening platform. The team there have been able to demonstrate a phenotype in some of the stem cells from people with sporadic MND. I think that's really interesting. It's still very, very early, but it's really interesting to see the potential of that. In Australia, we're also supporting the development of national clinical care guidelines for MND, which don't exist in Australia at the moment, so that's another exciting opportunity. But I think thinking more globally or more broadly, one of the things I'm really excited about is just the enthusiasm for collaboration. And I think we've seen that over the last 2 days, that people are really starting to think about things globally. And I think a lot of people are doing great work in their own countries or their own regions, but I think we're starting to see that shift of we all need to work together to really move the dial.
Well, what excites me in my region is just the fact that we're starting to mobilize and trying to change the everyday experience of people living with ALS in Africa. They've been neglected for so long. So that's exciting, and we hope to grow slowly, step by step.
Beck, you stole my line. How did you know? So bringing a global perspective, what I'm really excited about is really all this fostering of collaborations, as Beck said, just between today and yesterday and the next few days, because, this is going to be a long week for most of us, a very exciting long week. We'll have events that will really show the value of collaboration. And I think now more than ever, I did my PhD in Europe and my postdoc in the US. And more than 10 years ago, those two areas basically weren't talking to each other. So the fact that we are able to talk to each other about basic science and about clinical research is incredibly exciting now more than ever.
So I was trying to think about this, and I was like, what am I most excited about? And then I kind of came up just about half an hour ago with the idea that, so I'm thinking back 10 years to where we are today, and I look at, we talked about Fight MND and all the money that's been raised, and the Netherlands has been able to do incredible things in 1 week, raise 7 or 8 million euro, and the UK MND Research Institute, the CDMRP DoD program is $40 million US for research. ALS Association, MND Association, there's more money than ever, and this is incredible. And I just keep thinking, 10 years ago when Answer ALS started at 40 million, I thought, that's so much. And now we're finally getting in a realm where we're getting enough that we can do something, which is incredible in the field. And it's still not enough. We know it's not enough. So then it comes to what Fernando said yesterday from ALS Therapy Development Institute. I'm not sure if he's still here, and that's complementarity. When I look at the landscape, not only is there more money, but all these funds, each of the individuals are starting to look at how can we fit a specific niche in the field and work complementary to each other, which I think is really exciting. Again, we're just getting to the start of that, and where we're going to be in 5 years, I think is going to be really exciting. And then last to that, I would say that there's no small coincidence that this is in large part due to concerted efforts, and we're not there yet, to the field making space. I'm looking for Norm, I don't know if he's here, making space for valued advocacy of the community of people who are affected by ALS, because they are driving us to do that complementarity, and there's no voice that is more powerful to raise those funds that have come from those different areas than having them at the table in a more effective way than we have in the past. We still have a lot of work to do on all of these areas, but those three things combined make me very excited about where we can be in the next 5 years. And anyways, that's what I came up with.
Yeah, for me, I think the central dogma for neurodegenerative diseases has been that we can stop the disease or we can slow down the disease. And I think Khalsadi's data is showing us, and I think David said this earlier, that we can actually restore function. We're seeing that with patients reporting that on Khalsadi. What's really great about Khalsadi data, and Stephanie Fredette talked about this a little bit, is the real-world data are actually matching up with the double-blind study, and which is nice to see that now we have data in the real world that NFL is being reduced and that function may be restored. So, this whole idea of can we make ALS a livable disease? Yes, it's in a smaller population of SOD1 ALS, but it really tells us that the dogma of thinking about stopping or slowing the disease we can go beyond stopping and slowing. We could potentially restore function. And so that is something that's really exciting.
Excellent. And if we can turn to what you think the challenges are in our own areas, just briefly. I mean, there may be many, but let's just have a quick run down the line. Do you want to start, Kuldip?
Do you want to start at your end?
Yeah?
Yeah.
Beck?
I think there's lots of opportunities and challenges. I think one of the things that this group talks about a lot is around how we work together and what are the things that we go after to move the dial on. I think we hear a lot about data and big data, and it's how do we bring that together, and then what do we look for? So I think having the time to do that strategically and to have the funding to be able to prioritize projects like that, I think it's an opportunity more than a challenge that we can tackle some of those big questions.
Well, I could speak for a long time on challenges, but I guess the bottom line is that we as the low middle income countries, we do need help, and we need financial help. I was just saying to Cole that we can make a dollar go a long way, so it doesn't have to be big grants, but I would just put it out there that it would be, having a small pot of funds that the low middle income countries can compete for would be, I think, a good strategy.
So I think the challenge that I see, and this is going to be no surprise to anybody, is generally heterogeneity, and that is within the clinical population, but also as a basic scientist, it's somebody who worked with in vitro modeling, identifying that signal that the previous panel was talking about. We've made great progress, but it's still very, very hard to understand what that signal is. And so the challenge that I see is really heterogeneity in the population and in any prediction model.
And so one of the challenges, I will give a shout out to my colleagues, actually, Tammy and Elida, who put it much more eloquently than I was planning to now. But one of the things that we've talked about quite a bit is understanding that challenge right now between the urgency of the unmet need of the disease and that space where there seems to be a signal at a very small, underpowered clinical trial. We have a long history of being fooled by smaller, underpowered studies that turn out not to show efficacy in the end. And how do we figure that whole landscape out effectively and in the right way, collectively? And with that, the communications, and how do we balance the communications of what we know and what we don't know between the needs of industry to try to get investment for those trials and trying to help the community to understand what we really do know and understand from that data at that very limited stage before they've gotten to a pivotal trial. And I think that's a big challenge that we have to work out, and right now seems to be a particular hurdle for the scientific community in ALS. And I think Kuldip's gonna.
Yeah, and the challenge I'm seeing is kind of related to what David is talking about, which is right after AMX 0035, Albrioza, was approved both in Canada and US. You saw a lot of excitement in the venture community. There was, a lot of positivity. And then when the trial failed and the drug was taken off the market, we are seeing the pendulum swing the other way. And that is really concerning because, and we're hearing this directly from small biotechs that are coming to the association and saying, we do not have funding to take our programs forward. And something David said, companies are trying to balance that. How do they speak about their phase 1 and phase 2 data while still exciting the venture community so that they can continue to keep going? I think that's a real challenge because we can't have companies. ALS1 had a symposium a couple of weeks ago, and every company, one after another, was talking about efficacy with an N of 12, N of 8, N of 14, N of 30. And that cannot happen in our space. We cannot be talking about efficacy with small, underpowered trials. But I understand why these companies are doing it, because if they only focus on what phase 1 and phase 2 is supposed to be, which is dose selection, PK/PD, target engagement, biomarkers, maybe the VC community is not interested in funding them. And so, it's a balance. How do we scientifically on this stage make sure that companies are not talking up efficacy in underpowered trials, but yet how do we, on this stage, provide a message to the venture community that ALS is still really tractable, and hopefully use Qalsody as an example of that.
Yeah, thank you. So we'll open the floor to any questions you would have. Please raise your hands. And if not, we'll go to the online questions. Do you have anyone like a question in the room? One at the back there. Thank you.
Hey, thanks for your comments. I was wondering if any of you had some thoughts around heterogeneity. What is working to try to draw a representative population in the trials?
It's a good question. Who would like to take that? Dave?
Clarify? Yeah, you. Do you want to clarify that, Rob, a little bit in terms of specifically how heterogeneity is affecting clinical trial outcomes, and how do we do a better job of approaching that, or was it something else?
Earlier, with the ALS Focus survey, we saw 95% of respondents were white. How do we make sure that clinical trials enroll a representative set of participants so that we can make the most informed conclusions?
Do you want to go? We work harder to address that, and we have, when you point those things out, we go to the appropriate people in this room who can help us, I think, to do better at that, and making sure that we're reflecting on that and figuring that out. I personally don't have a specific answer, but I think that the more we talk about it, the more we're going to try to figure out and do it. And I think even things that, like Allison presented before and stuff, tells us we have a lot to learn on how to approach people to have a more balanced understanding of who wants to be in trials, how do we incorporate people into trials, and obviously earlier with the different networks, how do we get this around the world, and how do we understand the disease in different areas around the world and how treatments will be effective. I don't know if that answers the question, but I think Martina was also going to.
I don't know. Is there? Does anybody? Oh, the question is there an example of an inclusive clinical trial, even, I guess, in another discipline? It doesn't have to be ALS.
No, yeah, specifically in ALS. Like, I'm reacting to your comment about heterogeneity being the challenge. And I'm sorry, I'm with a pharmaceutical company, Ionis. So I'm asking this question from that perspective. There's a lot of pressure on us to enroll clinical trials as quickly as possible. So in one way, you're disincentivized to enroll a representative population if you prioritize speed. But we want to do the best experiment possible, which means to have a representative population. So I'm asking for some ideas maybe from this illustrious panel on how pharmaceutical companies or early drug developers can think about addressing this. It's a very important topic in a lot of different disease areas. In rare disease, it gets even harder.
Yeah, well, the association has tried out a couple of things, Rob. I think you know about these. One is we had funded trial sites to increase the sort of the DEI component of how they're going out and reaching out to communities. And I'll give you an example. We funded Henry Ford Clinic in Detroit, and this is a clinic that was predominantly seeing white patients in the middle of Detroit. And so the idea being that our funding can be used to navigate patients from underrepresented areas into both the care of the multidisciplinary clinics, but also for the trials that are being run out of those same clinics. The other way is, working with groups like My Tomorrows to navigate patients, again, through their doctors to trials. I don't know if there's an example of a trial where we have successfully gotten full, the type of diversity that we would like to see.
And just to clarify, are we talking about US specific trials, or is it a global?
Yeah, I think diversity, and I think it's agnostic to the country. I think there's a greater focus of it here in the US for sure. And some of this comes, there is additional pressure from regulators and payers on having clinical trials be representative. So that's where my mind is going, coming from a pharmaceutical standpoint. I appreciate the conversation. I don't want to hog the mic. Thank you for the examples, Kuldip, and happy to talk about this later.
Yeah, I think there's a lot of things we can talk about on that. And there's so many different layers of it. And also we're in Montreal, it's not here in the United States. I had to. I'm the token Canadian.
Thank you for the question.
Hi, I'm Matt Tilley from Fight MND. I want to ask a question about pharmaceutical companies, but I wonder if security could stand next to him first. I don't want to offend you. A question for the panel more generally, and you talked about funding, David, is that we obviously fund a lot of work across the whole ALS MND sector, but there's moments of jeopardy where we go, well, if we step in here, pharmaceutical companies are a little bit let off the hook, or are we creating a bad precedent where they start to think, okay, if we hold off long enough, funding entities will step in here and we're not so much responsible for it. And they're difficult decisions to make. I'm interested on, I guess, some sort of guidance or general thoughts from the panel around where do you think that responsibility lies?
In terms of funding trials, you're saying in terms of funding trials that maybe industry should be funding instead? Or, I mean, I think it's a problem we don't generally have in the field that much yet. And I think it's a great question to be having, to know that there is enough funding coming through certain philanthropic regions to be able to support clinical trials that are effective and getting to endpoints that are meaningful. Do we think, I mean, if we ever get to that stage, I think it's a good question, but globally, I'm not sure there are a lot of places where nonprofits are able to have enough funds to support supplanting. Even investigator-initiated trials are hard to fund sometimes in phase 3 level. So I don't know, I'll look down the panel as well in terms of, I think it's a great thing that we're at that space, but it's a good question that's leading to where we may get as I talk about us having more funding, and we need to consider it, absolutely.
Do you want to take anyone else want to add to that?
I mean, what do you,
Beck?
your fund?
I think probably Matt's question's probably about those early phase investigator-initiated trials. Okay. And then when they get to a stage where you're looking at progressing it through to the next phase. Even things like for investigator-led trials, then an open label extension type setting where should a not-for-profit be the one that's funding that, when if the drug is effective,
right,
they're not going to be the ones that are going to benefit.
Yeah, and I was going to say the same thing. I think for early stage, phase 1, phase 2, to de-risk the program, I think that's where the nonprofit funding comes in. But when we provide funding for those type of projects, we ask the companies, where are you going to get the next funding from for phase 3? Because nobody on this stage should be funding phase 3. We should have de-risked the program enough where the companies can now either get venture funding or partner up with, larger pharma to take that program forward. So.
Yeah, and it's also important that the landscape's still attractive to investors as well. So if we have a responsibility to make sure that things are done properly and communicated in the right way, and we don't need to be in the position where trials are failing all around us, or there's a difference between the way trials are conducted in different parts of the world. So I think it's really important the way that we present the M&D space as well to attract that investment. You know, I think that's a really important thing, the way the landscape is perceived.
And I just had, Nick, that maybe the UK is a really great place to have a chat, Matt, with, because, I mean, there's so much happening with repurposed therapies going into experts ALS, moving through to potentially the MND SMART trial, the experience that you've had with Miracals in terms of being an investigator-initiated large, long trial, and what happens at that end stage between that balance of how do you get this potentially considered through a regulatory pathway or access, and also considering the potential of needing an industry partner to be involved in that. And that's obviously a very complex situation right now, but I think those learnings could really help a group like Fight MND to help navigate those situations as well, don't you think? Or,
Yeah.
I don't know, I'm looking...
No, I agree. I think it's important, yeah.
Yeah.
Thanks. Is there a question of... Sorry. Oh, we'll go to this guy. I can't see much.
Does it work? Okay. I was jumping to the previous question. I'm sorry, but I would like to help in finding eventually an answer. Because learning from other disease in which we are working for diversity group inclusion, especially because from the pharma perspective and also from the regulatory agency perspective, they ask us to build clinical trial in which we could represent the most part of the population. And one of the points we have found around the world is the first point is the language barrier. So maybe could be really useful having the same maybe guidelines or something like that, or also the outcome measures translated in different languages. This is one point we saw that could maybe match the answer from the regulatory agencies, and maybe for the pharma. Could be maybe useful work together in that direction, maybe could be a really practical answer from your question. You're welcome.
Thank you.
Who I am? Okay, sorry. I'm Riccardo from Italy, especially from the ALS Italian Association, and now I will be a new member of the Alliance of ALS and MND. Okay.
Thank you. Thank you for your comment.
Hi. We have a question here. Marcela from Acela. I want to ask, what have we learned from previous trials that didn't succeed, and how is that shaping future research?
Yeah, really key question. You know, it's really important that we always learn from trials, and I think there's been many examples where we haven't learned, and we're still not learning, and that needs to be changed, whether it's target engagement or a biomarker that's measured in every single trial, a standardization across trials which are done in every different country. You know, I don't think we're in a really good place with that at all at the moment. And I think it's critical that every trial we learn something about the biology, we understand where it's gone wrong, or if it works, why it works. So, does anybody want to add a bit more to that, or?
I just, target engagement. We need to learn scientifically from the trials that we do, because it honors the participants that have taken their time and effort to be part of it. And if we've learned nothing from those trials, I think we need to push everyone who runs a trial to do their very best to do that, to make sure that we are not using that valuable, I don't even know how to say it appropriately, but doing that in a way that we're not learning and advancing the science, because I believe that's why people participate, is altruistically to drive the science forward.
We have a question here, Dave.
Thank you.
Hi, David Busak, Patient Fellow and ALS Network. I know there are lots of reasons, coming back to that question of diverse trials, there are a lot of reasons that make it hard to enroll people, but I think that Allison really hit on one, which is basic trust. I know in the US, at least in the Native American population and in the Black population, there's a huge amount of distrust. And in our current political environment in the US, there's a huge amount of distrust. So I'm sure there are similar things in other parts of the world, but how do we begin to overcome that distrust without getting Tricia, the trusted nurse or whatever her name was, to go to every household and say, you can trust us?
Yeah, I think that's a really important point. I mean, one of the things is that people don't often want to listen to experts. So, you know, if we've got patient fellows or ambassadors that are going out there and explaining communication in a way that brings that trust, I think that's really important. It's not an easy thing to do, but it's something that we should definitely be doing and should be doing more of. Called it.
Oh, go ahead.
No, you go.
The other issue is also that our multidisciplinary clinics look like that too, because the clinical trials are happening in those clinics, and that's where the patients are getting recruited from. So if the clinic is not representative, then the trial is not going to be representative. And so you're right, people are not, how do we get people to get to a multidisciplinary clinic so that then they can be participating in a trial? So I think it's both on the care and the trial side. Sorry, Beck.
Yeah, I was just going to sort of agree with what Nick had said, that we have sort of a more newly established lived experience network in Australia. And one of the sort of core focuses for us going forward is to have more diversity on that network. And we had a presentation from a woman with a First Nations woman in Australia whose dad died from ALS. And some of the things that she brought forward, including the fact that First Nations people are really against things like donating their brains or spinal cords to research because of the history of their culture and remains being taken overseas. We just didn't really even think about that. So I think if you don't have those voices talking about what those barriers are, then you won't be able to overcome them.
Thank you. Thank you for your question. Do you want just behind you?
Sorry.
Sorry, Kathy.
It's an off-the-wall question. If I gave you $100 million today, what would you do differently?
That's a nice question. Okay, let's take that in turn. Shall we? Do you want to go, Beck?
I have a whole plan.
Okay, go on, Dave. David?
I wouldn't do anything differently because I don't have $100 million. I mean, I think we all think about what could we do with a lot more. And I think it really comes down to the idea of, again, collaboration, complementarity, and having a bigger vision for what's needed in this disease and bringing together people from all parts of the ecosystem to talk about what those gaps are and addressing them effectively and together. And I think that 100 million will go very fast. And I'd like $1 billion. And we will spend it, it will be effective, and we will drive things forward faster. And I absolutely believe if we put the right people in the room and we give them the drive of what they need to do collectively, we are in a good place in this field to be able to do it. So if you can go out and raise $100 million, I think we're happy to help you figure out how to direct that.
I think also we've heard about financial limitation in different scales, right? Like everybody has financial limitations, but we've heard from Janine and from Elazam, from the Pact Health networks, that really there is an incredible need for investment in those areas. Also to discover heterogeneity and diversity, which we don't really understand now, right? So I think a large part of that 100 million will have to be invested in those areas to bring them up to speed in terms of facilities and resources with what's happening in the rest of the world. And at that point, we'll have a clearer picture, and we can make an informed decision on where to go.
Thank you. Are we good? Kathy?
Me?
Yes.
Okay, great, thanks. I still get really excited when I hear stories about tofersen and you know what its impacts are to the ALS MND community. And I just wonder, I know we don't have the entire SAC here or the entire world here, but just in terms of availability going forward and, you know, access to that for people that need it. Could you talk a little bit about that? And particularly, Nick, can you talk about what's just happened in the UK?
Yeah, so in terms of accessibility, I mean, that's, again, another area that is difficult. There's not even access for everybody everywhere. And even in developed places like the UK, the regulatory framework to allow people to get access is difficult. So in the case of tofersen, the authorities for people to get access through the national health system have assessed tofersen in the wrong way and have tried to assess it as if it works for everybody with MND rather than the around about 2% of people with an SOD1 gene change, which really shows a kind of naivety of understanding MND and what it is, and that it's different causes, different subsets of disease. So there's been a campaign and lots of work by many people, including the association, to try and get NICE to change its decision. And we've heard recently that they have changed their decision and they will now assess tofersen in the correct manner, we believe. That doesn't necessarily mean that people will get access to the drugs through the National Health, but at least it's a step forward that now Biogen can talk to the National Health Service to get access to people with MND for the drug. I have to say that Biogen have also been providing the drug to people free in the UK who were able to get it, but that couldn't go on indefinitely, so credit to them as well. But credit to people living with MND in the UK that got behind the campaign, filled in a petition, etc. But it shows that, you know, people can move the authorities and the regulators when we all work together, and that's credit to people living and affected by MND. But that's in the UK. You know, it's approved in Europe and the US, but in many other countries, you know, access is going to be very, very difficult indeed. If that answers your question, Kathy.
Yeah, and I was just going to add, obviously, the obvious point too is that if you need access to genetic testing and genetic counselling to even enroll it or be eligible for the therapy, so that's another big issue too.
Yeah. There's a lot of work to do. But the Alliance is, you know, shifting the dial across the world, I think, which is really key. I think we're probably short. We're out of time, are we? Have we got time for one more question? One more? Squeeze on in.
All right, one more. We're going to go back to the funding question, because I really want to take this gentleman up on his offer of $100 million. So here's really my question, though. We've talked about, David, you talked about target engagement, which leads me to believe that where we need to fund on the investment continuum is basic science. We've talked about a problem with VCs maybe removing themselves a bit from the space or being a little skittish, which tells me we need money later on in clinical development. Where do you see the biggest need when you look at the continuum of where this research needs to be funded?
Yeah, that's a great question. I mean, I would say, and it was touched on earlier, really, effective ways of measuring and understanding what targets we're looking for, target engagement and biomarkers, so that we can see whether drugs are working, we can see what happens, we can get these things to people earlier in diagnosis, so we know the drugs are getting where they need to be very early on, and we can't do that without effective biomarkers. So work needs to be done on that, absolutely, and is ongoing considerably as well at the moment. Would anyone like to add to that?
I'd just shout out that, you know, Fight MND held this global roundtable really to assess some of those gaps and bring together a group of people, and we'll continue to expand that out, hopefully with a large group of collaborators to understand what those gaps are. And they, in that group, came up with 7 key areas that they felt were really places we should be investing funds to try to move the needle as quickly as possible. And I think sort of what's come out of it is how do we shave decades off the disease in just a series of years. And I think that will be more widely put out there over the next several months. And right now it's sort of being workshopped, but I think that that will hopefully be partially a good answer to what you've asked.
Yeah, thank you. So I think we'll wrap that up there. But you know, just to say that we are all rowing in the same direction, and there's great things happening in motor neuron disease, ALS. It's not perfect by any means right now, but progress is really being made, and everybody's working really hard to try and find the answers in collaboration with people with motor neuron disease, ALS as well, pharmacy and industry as well. So you know, we'll keep moving forward with the fight. And again, our role as the council is really to try and digest and understand the information so that people can really understand what's happening on the landscape as well. So thank you very much to our panel, and thank you for you all for listening to us today. Thank you.
Thank you. That was fascinating. So I think it's exciting to see how much has been done, and it's also a lot of energy pulling up our sleeves to achieve what needs to be done. Okay, so this is the last part. I'd like to thank you for giving me the opportunity to be a moderator today. It was quite a privilege to listen to this, and it was very inspiring for me. So I took a lot out of that, and so thank you very much. So now I'm very, very happy and privileged to invite Karanit Ballas, CEO of the ALS Association and Chair of the Alliance's Board, to give us a sneak peek of what's coming up.
Sorry, I'm a little bit shorter than some who have come before me. Wow, what a wonderful couple of days. So first, I have the privilege of giving you a sneak peek as to what we're going to be doing in 2025. And for those of you who this was your first conference, I'm certain it won't be your last, right? You're gonna join us next year. So I think we've got a little video to show. It's gonna be a little different than Montreal. Leave your parka at home.
2 words that best describe the lifestyle and culture of San Diego, California are laid back. People from around the world travel here to relax, play, and experience the shimmering shoreline, the beautiful backcountry, and everything in between. There is nothing laid back and relaxed about the burgeoning biotech and life sciences industry here. San Diego has become a global hub for some of the most exciting work being done to find treatments and cures for the world's most devastating diseases. The ALS community welcomes you to America's finest city for the 36th Annual International Symposium on ALS MND, coming December 2025. Start making your plans now. The 36th Annual International Symposium on ALS MND in San Diego will be the perfect spot to see the sun set on this devastating disease.
All right, so I hope that you will join me back in the United States, my home country. We want to welcome you back. Now, some of you might be thinking, wait, weren't we just in San Diego like a couple years ago? Kind of, yes. But there was this thing called the pandemic. And so when that happened, there were a lot of different issues. It took us, 8 years to finally get here to Montreal, and now we're gonna be back in San Diego twice. So come on back, the weather's nice. Like I said, you can leave your parka at home. We're gonna have a really good time. With that, you are now part of this global community that you've already been a part of, whether you knew it or not. And there are a few things I wanted to highlight and bring your attention to. One is that we are going to be launching another fundamental rights survey. Our goal is to hit 3,000 participants, and we need your help to do this. Marcella, you said you were number 3. Colombia was number 3. I will tell you the United States was not not number 3. And so I really hope that you will help us, because we do need that diverse information, right, Rob? We need to figure out what's happening around the globe and what the needs are. In addition, you'll be getting a survey for this conference, and I want to make sure that you fill that out, because that helps us really hopefully bring even better information, content, and conversation for next year. So please fill that out. And then I just wanted to make a couple other comments. There are a lot of really interesting words that I heard over the last couple of days. Partnership, collaboration, focus, complementary. And I hope it's not lost on you, because it's certainly not lost on me, that sitting in this room for the last 2 days, as we work together, trying to make sure we're not duplicating efforts, trying to answer difficult questions and have real conversations, that while the global community seems, I would say, a little bit off its axis these days, when you look around at the 2020s and what's happening, and I know I don't need to go into detail, I am particularly heartened that this global community is not off its axis at all. We are very focused. And I'm so proud to be a part of a group of people that are so clearly focused on bringing forward a world without ALS and MND. So I personally would like to thank you for being a part of the conversation, being part of the solution, and being able to say no. As a global community, we can do a lot together, and we're gonna leave that other noise at the door. So thank you for that, and I just want a round of applause for this group of people. And that also includes our virtual community as well. I've been getting text messages last couple of days, and like, I don't feel like I'm there. I think Gujin sent an emoji, like he's crying that he's not here. It is not just in this room, it's all across the globe. So thank you. So that officially concludes our meeting. Excuse me, oh yes, I get to invite you. I get to invite you back into the foyer. From 5:30 to 7, you're gonna have a chance to get together oh, this is really cool, in a shared creative space. So this means we're gonna do some artwork together. There's gonna be music playing in the background that has been created by people who are living with ALS and MND, so tune into that. There's a lot of different tools so that people with all sorts of abilities, and by the way, I'm a terrible artist, so I'm not sure I'm actually gonna participate in any of this, but any kind of ability, we have some tools for that. And so I really hope that you'll join us and lean into that, because then we're going to collect all of that together and create kind of a tapestry of all of this different type of art that we're gonna do together. And with that, I'd like to also invite Laura. Where's Laura from the Les Turner Foundation? There you are. Come on up. So Les Turner Foundation has been a generous sponsor, a longtime partner, and I'd just like to bring up Laura to close us out and also join us in the next session. Thank you.
Thank you so much. I'm Laura Frevaletti, the CEO of the Les Turner ALS Foundation, and so honored to be here with all of you the past few days. I'm incredibly proud to represent the Les Turner ALS Foundation. It's been a cornerstone of the ALS community for nearly 5 decades. The foundation was started in 1977 by the family of Les Turner, who was diagnosed with ALS at the age of 36. This inspired them to raise funds to support research and provide critical resources for people living with ALS and their families. Just 2 years later, they established one of the first ALS research laboratories in the United States at Northwestern Medicine. Today, we continue to be a leader in the fight against ALS. Our educational programs, online support groups, and webinars reach audiences around the world. And our Les Turner ALS Center and Lois and Solia ALS Clinic at Northwestern Medicine is led by world-renowned scientists and clinicians, continuing to advance research into the causes, cures, and treatments for ALS. I came to the ALS community after losing my brother-in-law, Gary, to ALS in 2022, and I joined the foundation in early 2023. I'm a newcomer to this community, but I've been incredibly moved by the sense of community I've experienced. Through the Alliance and this broader network, I've met so many inspiring colleagues and allies who are working tirelessly to make a difference. What excites me most is a word that we have heard continually the last couple days, and that is the collaboration and the opportunity to create something together. The Les Turner ALS Foundation welcomes and are eager to be part of efforts that drive progress forward. Finally, I just want to highlight our deep commitment to the Alliance. The Les Turner ALS Foundation was a proud founding member in 1992, and we've been actively supporting the Alliance since then through the board, on committees, and other ways. And this year, we're thrilled to be a sponsor of the Day in the Life Suite and the art reception, which follows. And I did take a look at the Day in the Life Suite signups, and I don't think there were very many. So please go sign up and experience that. So before we all go to do a little art, whether we're good or not so good at that, I want to invite you to explore our brand new My ALS Decision tool in participating in clinical research and our corresponding PDF guide, which we developed in partnership with Niels. These and other resources for everyone treating ALS, investigating ALS, or living with ALS can be found at lesturnерals.org. So thank you again for the work that we all do in this community, for our continued fight and our partnership and our collaboration. So let's go make some good art. Thank you. Have a nice evening.